Hey guys, welcome to my channel. This is Omolo the Great. If you're new to this channel, remember, this is your physiology store where physiology concepts are made super easy to understand. All you have to do is hit the subscribe button below, like and share this video. So today we are going to talk about the transmembrane transport, or what we may call the transport across the cell membrane. Cells usually do a lot of metabolic processes or metabolic roles, and they require a lot of nutrients in which they have to utilize to generate energy or synthesize other molecules. So these nutrients, they can obtain them from the extracellular compartment by obtaining them through the transmembrane transport, through different mechanisms through which these nutrients actually move from outside the cell into the cell. Similarly, after the cell has done its metabolism, it has several waste products that it can actually pass out from its intracellular compartment and send them out to the extracellular, or to send them out to be excreted or transported in blood to excretory tissues. So these processes through which these molecules move from one area to the other of the cell, either from inside the cell to the outside of the cell, or from outside the cell to inside of the cell, are the mechanisms we are going to look at today. And these mechanisms are the mechanisms of transmembrane transport. So broadly, we can categorize these mechanisms into two. So there are two forms of transport that take place across the cell membrane. One is what we call the passive transport, and also we have what we call active transport. Okay, so the transmembrane transport mechanisms. So transport can either be passive mode of transport or active form of transport. Under passive form of transport, we have different forms. We can have what we call diffusion. And then there's another form, what we call osmosis. And then the other form, under active transport, we can have primary active transport, and then we can have secondary active transport. Yes, and then under diffusion, we can have simple diffusion, and then we can also have facilitated diffusion. So under the primary active transport, we shall look at it. Then under secondary active transport, we have what we call the core transport mechanism, and then we have what we call the counter transport mechanism. Okay, so this is a broad classification of transmembrane transport. So if you know and have all this at the back of your mind, then you have the whole idea or package of transport of molecules across the cell membrane. So briefly, to start to highlight, I'll start with a comparison between passive and active. So passive transport, this is a form of transport that takes place across the cell membrane without requiring any form of energy. Energy in the body is usually in the form of ATP. So this passive transport, there is no energy expenditure compared to what we call the active transport, where we have energy expenditure. So in passive, we are saying we have no energy expenditure. So no ATP is involved. So no ATP breaking is involved. Whereas in active transport, we have energy that is broken down, or we are expending energy, what we are calling energy expenditure. So this is one most important distinction between passive and active transport. You should remember this. Passive transport, to repeat, is a form of movement of molecules across the cell membrane that does not require energy, or where there is no energy expenditure. Whereas active transport is where we have energy is expended, or where ATP is cleaved to release energy in form to release energy that is utilized in catalyzing the process of transport. So we have talked about, we've broadly classified our transport, membrane transport, into passive and active. And I've told you passive is transport that does not require energy expenditure, whereas active is mode of transport that requires energy expenditure. So as we have already said, passive has two forms. We have the osmosis and the diffusion. So for osmosis, this is a process that involves movement of molecules from a region of higher concentration to a region of lower concentration across a semi-permeable membrane, or a selectively permeable membrane. And an example is actually the water, that actually water molecules that can easily move across this semi-permeable membrane. So for diffusion, this is where we're going to spend much more time. So under diffusion, so diffusion, this usually, one thing you should remember, molecules are usually under a kinetic motion. Molecules in air, for example, right now in this air, right now, we have molecules that are actually in motion, but I cannot recognize or see with my eyes. But so in diffusion, molecules have an energy that they are utilizing. There's a passive form of energy. So they are usually in motion, or they are usually moving. So molecules in the under diffusion, molecules or kinetic molecules are moving from a region of higher concentration to a region of lower concentration without need of energy. Okay, so for example, if you look at this illustration here that you can see, usually this is seen in kindergarten schools, where it's usually used for playing by those young children. So we have our young girl here, okay, who desires to play. So one thing you should remember, these young girls love to play, okay, or although it's a boy, so we can use a boy, okay. Boys are quite usually inquisitive and want to know how things work, and they're able to try out a few things here and there. So this, just like they're very active, that's the form of molecules I'm talking about. Molecules are always in kinetic motion. So this young girl loves to play, and this young boy, he decides to climb on the stairs, this few, and he is right here, okay. He is right here, and he is ready to slide off. So he sits down, okay. He sits down, and he is ready to slide down. So, and he is going to move right away from up here, to move up to down here, and eventually he is going to be here, okay, seated here, okay, where he has come, and then he will get up and walk back, okay, for another round. So how this boy is moving, when he reaches at the top of this, he is going to just slide over. He usually just slides, okay. So this sliding bit, the inclination of this usually slides at ease without any effort. And so similarly, molecules in diffusion that are always in a kinetic matter will move from a region that is higher to a region that is lower, down a gradient, okay. So this movement is very simple, does not require any form of energy, and this is what schematically I would use to illustrate an understanding of diffusion. So same thing happens if let's use actually now let's use the actual cell. So if this is a cell, for example, okay, a schematic representation or diagram of the cell with the fenestrated membrane. This is the inside, this is the outside, and this is the inside of the cell. Okay, so we have different molecules. We're having different molecules here, molecules of a substance, say water, for example. Okay, so these molecules, they are highly concentrated here. So for example, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There are about 10 molecules here, highly concentrated. So these molecules will move to the region of low concentration. Because they're always in kinetic motion, they'll move and collide with the walls. And when they collide with these powers along the walls, they're able to move on the other side. Okay? Eventually, the number of molecules on the side increases until there's a balance that comes, there's an establishment of a balance. Okay, so this is simply molecules are moving by simple diffusion. So how is this? Let's represent it on a cell membrane. Okay, so if this is our cell membrane, remember we said in the previous video, please, if you haven't watched the previous video on cell physiology, visit. It will let you know about how the structure of the cell membrane. We already said that the cell membrane is a lipid bilayer. Okay, I'm just trying to represent the lipid bilayer here. And so this is the lipid bilayer. Okay, so in this lipid bilayer we have some proteins that span through the membrane of the cell, and these are what we are calling our integral proteins. Okay, so let me give it a power. It's going to be this is one protein. Okay, so this is our integral protein, and as you can see, to continue this, our different layers, our lipid bilayer, okay, that has phosphate heads and hydrophobic tails. So usually in simple diffusion, molecules are going to move kinetically without any aid of energy, okay, or without need of energy. And usually these molecules in simple diffusion, they're going to move through these intermembrane spaces, that is if they are lipid soluble, or they'll move through with the help of a channel. Okay, so for example, there are molecules, and we are talking about simple diffusion. Okay, so simple diffusion, this will occur, movement of these molecules from a region of its higher concentration to a region of its lower concentration across this membrane. And this usually these molecules that can move through these intermembrane spaces, definitely these molecules have to be lipid soluble. So molecules that easily diffuse simply through this membrane across, they are lipid soluble molecules. So that is one form of simple diffusion. So one form of simple diffusion is movement of molecules through intermembrane spaces. That is by the fact that they are lipid soluble. So for example, here examples we can have carbon dioxide, some gases, we can have oxygen, we can have nitrogen, we can have also alcohol. Alcohol also does a lot. It can easily cross the cell membrane, and that is why I told you about in one of our videos, our previous video. Actually, alcohol, when we shall look in GIT physiology, when you take alcohol, it is easily absorbed within the stomach because it can easily cross the cell membrane of the cells because it is lipid soluble. The other form of simple diffusion is what we are calling this is a channel protein. Okay, a channel protein. Okay, so the biggest the other form of diffusion is movement of molecules through a channel protein. Okay, so the biggest difference between simple and facilitated diffusion is that simple diffusion does not require a carrier protein. So under simple diffusion, we know carrier protein, okay? Whereas in facilitated diffusion, you need to have a carrier protein. And I'm going to expand much about carrier protein when we reach there. So you need to have a carrier protein facilitated and then no carrier protein in simple diffusion. So what are some of the molecules that can actually move through these channel proteins? So for example, under this, in this we can have molecules, especially ions, that can move. We can have sodium, we can have potassium, we can have calcium, we can have hydrogen, calcium, name them, the chloride, so, and these other ions, okay, that can actually cross, and even bicarbonates, they can cross this cell membrane by the help of these channels, and that is a form of simple diffusion. So I need to tell you more about what we call these channels in simple diffusion, okay. These channels in simple diffusion, channels can be, for example, we have an example, we can have leaky, you've heard of leaky channels, which leaky channels could be, one, we can have leaky sodium channels, we can also have leaky potassium channels, but also another important thing to remember that we can help us classify these channels is what we are calling the gated channels. Gated channels. These gated channels can either be, we can have a voltage gated channel, but we can also have what we call a ligand gated channel. A ligand gated channel. Okay, so let me tell you more about the structural arrangement or the structure of these channels, because this is important to understand the details of transmembrane transport, especially as we're talking about the form of simple diffusion, and we're talking about the ions that actually utilize the channels to move across the membrane. So let's start with, let's talk about this potassium and sodium leaky channel. These channels here, one thing you should remember, sodium and potassium. Potassium is structurally bigger than sodium. So in size, in diameter, potassium has a much bigger diameter than sodium. So they across the membrane, we have, let me make it a little bigger. Across the membrane, the channel, the potassium channel structure, it is tetrameric, so it has 4 subunits. So in its tetrameric structure, let me illustrate here. This is one subunit, it has another subunit, and then it has a third subunit, and then it has a 4th subunit. This is just structurally to show you, and this is our cell membrane, okay. This is our cell membrane. This is our cell membrane. I'll always show you the lipid bilayer. This is our cell membrane. So these channels, for example, potassium, another unique thing. So I said it has 1, 2, 3, and 4. It has 1, 2, 3, and 4 subunits. And so these subunits have something what we call selectivity filters. We call them selectivity filters. Selectivity filters. So these selectivity filters are quite very important. Why? Because they are important. They help to determine the specificity or the selectivity of a channel for a given molecule. So this means that a potassium channel will be able to transport potassium more compared to the ability to transport sodium because of the presence of these selectivity channels. And what do? To make things easy for you to understand, if you are going to move through this this is like under the somewhere under the bridge, or maybe a tube, okay, a tube that you're going to move through, okay. So if you're going to move through this tube, we have someone that is within, okay. We have someone within, okay, that can aid you to move through across the tube. So if A wants to get across this molecule, okay, and so these, they are like, let me call them hands, okay. Those selectivity filters are like hands. So they are hands that are lying. If this is the tube, okay. So if this is the tube, oh yes, this is the tube. So within, if you can see, so in here there are some finger-like, finger-like projections, and those are what you call selectivity filters, okay. So this is my tube, and you're having finger-like structures that are within, and those structures there, what is their role? Is to hold the molecule that wants to get in, okay, and then strip it naked, okay. After stripping it naked, then they can transport you. So the only way you can move through this channel from anyway molecule A can move through this channel, molecule A has to be stripped free of its clothes and then sent through the channel. That is the only way, which means if you are not naked or stripped of your clothes, you cannot go through that channel, okay. And this is what means under normal circumstances. If we go back to our potassium, under normal circumstances, potassium, these molecules, when they are outside the cell, okay, this is the inside, this is the outside, inside. When molecules are outside the cell, they interact with different molecules of water, okay, molecules of water, H2O, 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 H2O. So this is a hydrated molecule, this one here. So this hydrated molecule wants to get in through its potassium channel, okay. So this hydrated molecule, let me call it a box here of potassium. As it wants to get through this molecule, it is first stripped naked, or it is now dehydrated. So it is made free. The selectivity filters make it to lose its water molecules, so that we have a dehydrated potassium molecule that is now going to pass through the channel. So this hydrated potassium molecule, this one, let me use a different color so you can see it. So this potassium molecule that is now dehydrated is now able to move through this filter to the inside of the cell, okay. So this is the one most important function of those selective filters. They free the water molecules from the potassium ions, and then it is able to move through the channel. And so if for example sodium wanted to pass through this channel, sodium will find some difficulty. Why? I told you already, the size of sodium is quite smaller to that of potassium, implying that as sodium is trying to move through this channel, sodium will not have an association with the selectivity filters. Just like you're moving through and the selectivity filters cannot touch you across, so they cannot strip you naked. So which means if you're not naked, you will not go through the channel. I hope this is quite very simple and clear. So if you cannot go through the channel because you have clothes, and you cannot be stripped because the selectivity filters cannot touch you, so you cannot go through the channel. So that is why sodium finds difficulty moving through these potassium channels. And this is why you also remember that at resting membrane potential, or when the cell is at rest, usually there is more movement of potassium ions than sodium ions, because we know there is presence of the potassium leakage channels, implying that when the cell is at rest, it is less permeable to sodium ions than it is permeable to potassium ions. And this, what I've just talked about, explains everything about that concept. All right. So we are done with the sodium and potassium channels. And then, so maybe one more thing. There are some factors that will determine the selectivity of the channel to a given ion that it will transport. And what are those factors? One, I've already listed. We've already talked about some of them. One, the size, or what we call the diameter of the molecule. Okay, we talked about sodium and potassium, that these channels are quite specific to a given diameter of a molecule. So if a molecule is smaller, or if a molecule is bigger and it is not corresponding to the size that the channel requires, that molecule will not go through. Okay. Two, the charge across the membrane. So the charge difference. I'm going to tell you about this. And then also the chemical bonds that are within the channel. The chemical bonds within the channel. And then lastly, also the shape of the molecule. So this molecule, if the molecule cannot sit or correspond to the size of the channel, then it will not go through. Similarly, I talked about diameter. So the charge difference, to illustrate, if this is our membrane, okay, I hope you can see it. So if this is our membrane, and this side, this is the inside, and this is the outside. So in the inside, we have a lot of electronegativity, okay, compared to the outside, okay. So let's have a charge difference that is, at this point, we're just looking at the negative ions. We have more negative ions on this side compared to the ions that are on the other side, okay. So if I introduce a positive charge, on the other side of the membrane, okay, and then I introduce a negative charge on this other side of the membrane. What is it going to do? The positive charge is going to attract all the negative ions, going to attract negative ions towards this other side of the membrane. So you find on this other side now, the membrane is going to have more of the negative ions. But also this negative charge plays a big role, because it is going to repel all these negative ions to the other side. So you realize that actually the charge across the membrane will depict or determine which direction these ions are going to move. Are we together? So I think that makes very good sense. And also the chemical bonds I talked about, you see these selectivity filters, it is a complexion of different chemical bonds, okay. So there are some carbonyl groups that play a big role in the dehydration or removing the water molecules. And so if this interaction of these chemical bonds is not really compatible for that ion, then that will affect the movement of that ion. So I think this summarizes all about the leaky channels. Then I'm going to talk about the gated channels. So we said these gated channels can be voltage gated channels, or they can also be ligand gated channels, implying that voltage, whether they are voltage gated or ligand gated, which mean these channels, they are conditioned to open. So just like if you come back at home and you're driving and then you reach the gate, then you hoot the car. So the condition for the gate man to realize that actually there is need to open the gate, the condition is hooting. Okay, you hooting the car. So when the gate man listens or hears the hoot or the sound of hooting, then here is actually, that's a signal for opening the gate. So same thing here. When the channels, these channels that are gated, which means the channels have gates, when the voltage changes and it is appropriate for it's appropriate or is able to be heard, just like the gateman hears the hoot, then the channels can be where the gates can actually be opened. So the condition in these two, one is a voltage that reaches a threshold or a voltage that is required to open a certain gate. For example, we said when you look at the voltage gated channels, we have sodium voltage gated channels and we have also potassium voltage gated channels. So these channels will only open only if a certain voltage that is reached, that is required to cause opening of these channels. That is when these channels will open. And for the ligand gated, so and a very good example of the ligand gated channels is the one of the acetylcholine. Okay, if you looked at the cell neuromuscular junction, we have a lot of different, we shall look at this when we talk about next in our subsequent videos on excitable tissues. But acetylcholine is released from these vesicles, and when it reaches the end plate at the neuromuscular junction, it is going to cause, if these are the channels, let me magnify that. So if this is, I'm using a different color. So if this is the channel, okay, this is the channel. It has a gate. This is the channel on the membrane. Okay, so and this is our gate. This is our gate. Okay, this is our gate. So this molecule of acetylcholine comes and binds here. So when the molecule of acetylcholine binds, it is going to cause opening of the gate, okay, opening of the channel. So the ligand here is acetylcholine. It is going to cause this gate to open so that we can have an influx. So if this is our gate, this is step A, and our gate here with the bound acetylcholine, and then this is our channel. Okay, so our gate, it has a bound molecule of acetylcholine. It is going to cause our gate to open. Okay, so when our gate is open, so now molecules can easily move, maybe sodium or maybe potassium. Okay, so these molecules can now move because the gate is open. So this is literally some to show you about the ligand gated channels. So the condition for the ligand gated channel is, for example, the one of acetylcholine is the binding of acetylcholine that is going to cause opening of the gates of these channels, of these ion channels. Then we get an influx of these ions into the cell. So that is one. So it's almost same thing happens for the voltage gated channels. The only distinction in these channels is that when you look at the voltage gated sodium channels, if this is our cell membrane, okay, and then we have our channel here, okay, so this channel has, okay, so if you look at this channel, okay, this is our sodium voltage gated channel. So our sodium voltage gated channel, when the membrane potential of the cell changes from, maybe in skeletal muscles, from -90 and is moving, becoming less negative to around maybe -55 millivolts, okay, this is going to cause opening. Okay, so under normal circumstances, our channel, sodium channel, has two gates. It has this gate and this gate. Okay, so this gate is the activation gate. The activation gate. And then this gate is the inactivation gate. Okay, so this channel, when the membrane potential changes from the resting state to less negative, it reaches a voltage potential or a voltage of maybe around negative 55 millivolts that is going to cause these channels to open. So this voltage leads to, it's going to cause this channel, okay, let me put it here. So this is the very channel I'm drawing here. So it's going to cause the activation gate, okay, to open. Okay, so usually these are the two gates, but you find under normal circumstances, you find this gate is at resting state. This gate is actually what? The inactivation is open. Okay, so when the potential reaches about negative 55, it's going to cause the activation gate to open. And when this activation gate opens, you're going to have a rapid influx of sodium ions into the cell. And this is why, as we shall see during the discussion on action potential, the rapid influx of sodium ions will then cause the phase of depolarization. So in this, one thing to remember about this channel, uniquely about this sodium channel, these gates, they open very fast and close very fast. So this shift or swift change in the membrane potential from this to this is going to cause a very fast opening of this activation gate, causing an influx of sodium ions. And then when the membrane, when the potential or the action potential has reached the peak, as we shall see in our next discussion, you find the inactivation, usually they say the channels of sodium close, then we start having opening of channels of potassium. But this is what happens. So when the channel, this is stage A, this is at rest, okay, and then here, this is when the membrane is depolarizing. So the potential is becoming more and more less negative to positive at around maybe 30 millivolts. Okay, so this so the next stage, so when the action potential has reached the peak, what is going to happen? Okay, what is going to happen? This is what is going to happen. We said, so this gate was open. So when the membrane potential reaches the peak, what happens to the gate? So this inactivation gate closes, and when the inactivation gate has closed, you cannot have any more sodium. Okay, no more sodium. Sodium cannot get in because this gate is actually locked. Okay, so when the gate is locked, no more sodium is coming in. So this is what happens. This is the value or the function or the code of operation of these different channels. Remember, they have the activation and the inactivation gate. Sodium has the activation gate and an inactivation gate. It's the activation gate that is open during at rest, that opens when the membrane potential changes from negative 90 to negative about 55, causing influx of sodium ions. And when the membrane reaches the peak, when the membrane potential or the action potential has reached its peak, then the inactivation gate actually closes to stop any more influx of sodium ions. So usually we say at the peak of the potential, these potassium ions then open so that the membrane can be restored to its resting state. So let me draw for you the potassium channel. Okay. So the potassium channel, for the potassium channels, they have one gate. Okay, that's the difference. They have one gate. Okay, and this gate is inside within the cell. So usually this gate, when the under normal circumstances, this gate is actually sometimes actually closed. So, but when the potential this side of the cell has become more electropositive, so the membrane needs to restore its electronegativity, that is its resting membrane potential. What happens? This channel actually opens, okay, this voltage gated channel. At the peak of that voltage, when the sodium voltage gated channels close, then the potassium voltage gated channels actually begin to open. But the only difference between this these two channels is that the sodium channels, as I've already said, they open very fast, whereas these voltage gated potassium channels will open slowly. Okay, so this channel opens and then we have an efflux of potassium ions. So that's what this is all about, these channels: the ligand gated channel, the voltage gated channels, and as already talked about, the leaky channel. So this brings me to the end of simple diffusion. Let's look at facilitated diffusion. So under facilitated diffusion, we say this is the movement of molecules across the cell membrane with the aid of a carrier protein. With the aid of a carrier protein, which means someone needs assistance or help to move through the cell membrane. So for example, if this is our cell membrane and we have our channels, okay, so this channel here, these are our carrier protein. Okay, so a molecule here requires to be helped to move by this carrier protein. So the unique property or unique thing about how these actually do this is that when this molecule actually binds onto this channel, the channel undergoes a conformational change. Okay, this channel will undergo a conformational change and it is be aided to move to the other side of the membrane. Okay, so this is an example of facilitated diffusion. One, we can have these what we call aquaporins, and then these are typically water channels. Okay, so these are being so studied, they're being studied so widely. For it, we can have, you've had aquaporins 2, 3, 4. These will come out more in your when we're talking about renal physiology. We also have urea transporters, which are also more prominent when you're talking about in renal physiology. So these are some of the examples of facilitated diffusion. So the molecule binds onto the carrier protein and undergoes a conformational change until deliver that molecule on the other side of the membrane. That's what you should actually remember. But in a nutshell, everything about simple diffusion does not require energy. Okay, and still facilitated diffusion will also not require energy, and both of these mechanisms will not require energy. Simple diffusion will have no carrier protein that is engaged, and for facilitated diffusion you will have a carrier protein that will be helping in the movement of this molecule from one side of the membrane to the other. So let's look at active transport. Active transport, we said this is a mode of transport that actually requires the expenditure of energy. And so it's categorized primarily. We said we have primary active transport and then we have secondary active transport. So let's focus much here. So primary active transport, this is a mode of transport that involves direct cleavage of energy, or ATP. So here we have direct breakdown of energy, breakdown of ATP. Okay, so there's direct breakdown of ATP, whereas secondary uses an indirect source of energy. Indirect source of energy. So when we look at let's look at the primary. So when we look at the primary form of active transport, primary form of active transport. So, for example, we have the sodium potassium pump, which you've already probably met. We also have what we have a hydrogen ATPase pump. We also have we can have a calcium ATPase pump. Okay, and one other common one here is the sarcoplasmic endoplasmic reticulum calcium ATPase channel that we shall be seeing more in when we talk about our muscles, when you reach muscle contraction. So one that has been widely studied is actually the sodium potassium pump. let me talk about this one and how it does its role. Structurally, this sodium potassium pump has two subunits. It has an alpha and a beta subunit. Okay, this is the alpha, rather the beta, and this is the alpha. So the two different roles of these subunits. One, this alpha, rather the beta subunit, it helps to anchor this channel onto the cell membrane. Okay, so it helps to anchor this channel into the cell membrane. So this is one most important role. And then this alpha subunit is actually a functional protein. So it plays a functional role, which will be mainly the movement, as we shall see, but also it further has something very important called an ATPase. It has an ATPase enzyme. And what does this enzyme do? This enzyme also just plays a role to cleave. This enzyme cleaves ATP to ADP plus an inorganic phosphate with the release of what? Of the energy. Okay, and so it is this energy that actually binds these, that actually is used in movement of these ions. So let me make this a little bigger for this molecule of this alpha subunit. So there's something more about this alpha subunit. This alpha subunit has two pockets or binding sites. Okay, let me two binding sites on the outside, and then it has three binding sites on the inside. Okay, so these binding sites that are on the outside, these are binding sites for potassium, and then these are binding sites, this one, this one, this one, these three are for sodium. Okay, so these are binding sites for sodium and these are binding sites for potassium. So under normal circumstances, when this molecule binds, let me draw it here. So when this channel has bound, okay, when it binds, we have 1, 2, potassium, and then it is binding 3 of sodium, 1, 2, 3 of sodium. When it binds 3 molecules of sodium that are inside the cell, and this is outside, 2 of potassium outside and 3 of sodium inside. So this causes an activation. It activates the enzyme. That enzyme is what we are calling the ATPase enzyme. Okay, this ATPase enzyme. When this enzyme is activated, it now cleaves this molecule of ATP to the ADP plus the inorganic phosphate and to release energy. Okay, and this energy is utilized to cause a conformational change, and this conformational change is what is going to result into the movement of this molecule. Okay, we're going to move the 3 sodium outside and the 2 potassium inside. So this is how this molecule, this channel actually works. This under the primary active transport. This is an example of the sodium potassium pump, or the sodium potassium ATPase pump. And so I've talked about the structure and its binding sites. And so the most important is the ATPase, which actually cleaves the ATP to release the energy that is required. And so this is employed still to the hydrogen ATPase, calcium ATPase, and more, as we shall see. The close example, this Seca pump in the muscle contraction. So then we have secondary active transport. So secondary active transport, it utilizes energy from another source or an indirect source of energy. So what happens here? But there are two forms. We have co-transport. Co-transport means you're moving together. Okay, you are moving together. So molecules, if this is the membrane, molecule A and molecule B, okay, 2 molecules are bound by this channel and they move together on the other side of the channel. Okay, so they are moving together. So these channels that play a role in co-transport, they move molecules, at least two molecules, more than one molecule are bound and moved on the other side of the molecule without a direct source of energy. So this energy is actually obtained from from somewhere else. And then for the counter transport, molecule A and molecule B are binding and they are moving in opposite direction. Molecule A is moving inside the cell, whereas molecule B is moving outside the cell. And so this is what it means. So it's about the core moving together and then the counter moving in opposite direction. So let's put some science and sense to this. Examples. This is core transport. Okay. So we have molecules that move along with sodium. We have a sodium glucose transporters. These are the most widely studied. There are different classes, different types. So you can have one, the others two. So this transport both sodium and glucose. Okay, so these two transporters will bind molecule of sodium and the molecule of glucose and transport it together in one direction. We shall meet this as we discuss more systemic physiology. Similarly, when we talk about counter transport, here we have channels. For example, we have the GLUT. So for counter transport, an example you can have, we have the hydrogen potassium exchanger. Okay, so this hydrogen potassium exchanger is an example. You can also have the calcium sodium exchanger, or there's also called antiporter or symporter. So this is an antiporter, this is a symporter. Okay, so these move molecules in opposite direction. But let me show you diagram schematically how actually these usually occur, because this brings a lot of confusion to many students on how these actually take place. So if this is our cell, the concept of the energy usually causes confusion. So this is, for example, the lumen, okay, and this is the basolateral side of our cell. So we have our sodium potassium pump here, okay, that's pumping 3 sodium out and then 2 potassium inside. Okay, so if you look at this, eventually you're going to have more of sodium that is outside the cell and less sodium inside the cell. So already there is a gradient where we have more sodium that is existing on the side than within the cell. So this gradient tends to create an energy that is going to cause, that will depict, we have also sodium here. So because now the concentration of sodium here is higher than the concentration of sodium inside the cell, sodium tends to move into the cell, okay? And why is this gradient has been created, the gradient enabling the movement of sodium from this lumen into the cell, that gradient has been created by the activity of this sodium potassium pump. So that's why they say secondary active transport occurs in presence of the primary active transport, because the energy generated is from the concentration or the electrical gradient that is created by this sodium potassium pump. Okay, so it's going, because there is a gradient of sodium that has been created, sodium is able to move from outside into the inside of the cell. But what is surprising, sodium does not move alone. Sodium, it will use, because of the channels we've talked about, this core channel, for example, core transport, this sodium will bind another molecule of glucose. So as sodium is moving inside the cell, it's also going to move this molecule of glucose inside the cell. So this is an example of co-transport. Energy utilized for movement of this molecule of glucose is generated by the gradient created by this sodium-potassium pump, which is a primary active transport mechanism. Okay. Whereas if it is counter transport, same thing. The energy gradient that is created in this case, we will create the concentration gradient that is required to move any molecule on the other side. Okay, so this brings out very well the idea of this transport, membrane transport, and we have highlighted all the examples on our membrane transport. So all you have to do is review this video and understand. If you have any questions, please put them in the comment section. If you haven't understood some area, put in the comment section and I'll be able to explain things better for you. Otherwise, thank you so much for watching. All you have to do is subscribe, share, and like this video. Thank you. Bye-bye.